Hello, this is Tommy. Welcome back to Chatnomics. In today's video, I'm going to show you how to do bulk RNA sequencing analysis from end to end. I'm also going to show you a trick that can help you to avoid repetitions of your R code, the powerful map function from the purrr package. And you can use that package to apply the same function to multiple comparisons. So the data set we are going to use is from Geo. This is actually here. So in this experiment, they have SW colon cancer cell lines under different conditions here. So we have some guide RNA control. So those are control guide RNA, but they also have two knock conditions for those two genes, ITPR3 and RELB. And those cells are treated under two conditions, normoxia condition and hypoxia condition. So hypoxia means low oxygen condition. Okay, let's go to RStudio and then do that from the very beginning, downloading the raw counts. By the way, I will have the link of this R Markdown files in the description of this video so you can follow along. Okay. So you can download those raw counts by following the information in here, this link here. You can use wget function and in commands, and then you do this wget, and then this is the raw count matrix that we're going to use. So essentially just rows are genes, columns are samples of different conditions. And if you don't have wget, you can use the curl command. You can just copy paste from Markdown files. Okay, let's just go through a standard DSeq2 workflow for a single comparison here, okay? So let's load the libraries. Okay. Okay, now let's read in the raw counts. Okay, so this is the file we downloaded. And you can use the read_tsv function from readr and to read in that count matrix. So this URL will be different from mine, like if you download into a different directory or folder. And let's take a look of the file. This is the first 6 rows of the file. As you can see, so we have multiple columns here. And the first column is the gene name, and then the rest are the sample IDs. Okay. So you can use the column name function to look at all the columns here. So the first column is gene, and then we have 12 different samples here. Okay. So as I mentioned, those are the different conditions: control guide RNA and knockout for those two genes, and the two treatment conditions, low oxygen condition, hypoxia, and normal oxygen condition, which is normoxia. Okay, so we will use DSeq2 for differential gene expression, and we need to make a count matrix from that data frame. Okay, so we can just subset this data frame and remove the first column here and turn that as a matrix. So let's do that. So this matrix has no row names, right? Just the columns, the samples. So now we need to add back the row names, which is the first column, which is the gene names. Okay, we do that. And let's look at it again. So now we get a matrix with rows are genes, columns are samples. Okay, let's do a differential gene expression analysis comparing hypoxia condition versus normoxia condition and use guide RNA, so there's no any knockout for the genes. Okay, so we need to first subset the count matrix to, so this is the normoxia condition for the control SG guide RNA, there's 2, and then 7 and 8, which are 1, 2, 6, 7, 8, and so this is a sample also control sgRNA, and then on the hypoxia condition. Okay, so you have 2 samples for each. So let's subset that and look at the subsetted count matrix. And now we need to make a data frame showing what are the experimental design. So we do that, a data frame function, and then make sure the row names of that data frame is the same column names of the raw count matrix. And if we do take a look, so this is like the design data frame. Okay, so we know those 2 are normoxia condition and those 2 are hypoxia condition. Now we can make a DSIG2 object. First, we can actually verify whether the column names of the raw count matrix is the same as the row names of that data frame here. Okay, so before that, we will actually filter the raw count matrix a little bit. So in this case, we are requiring 10 counts in at least 2 samples. Okay, so okay, and now we can use this index. This is true or false. So you will see that this many numbers filter. So we can do that. Now just follow the standard DSIG2 workflow. So make a DSIG2 object to data set from matrix. And then if you look at DDS, so this is a DSIG data set object. Now you can just do DSIG. This is the main function. It would estimating the size factors, dispersions, and also just normalize the data. Okay. Now you can use the results function to call the differentially expressed genes. And you can specify the contrast, which is in the condition column, comparing hypoxia versus normoxia. Let's do that. If you look at the results, this differential expression table, okay, it tells you conditions hypoxia versus normoxia. This is the number of genes that we have, and this is the same after we filter those low counts genes. Okay, we can actually define significant genes by using some cutoffs. So first we need to actually convert that as a data frame and then filter using p-adjustment value. For example, this column smaller than 0.01 and absolute log2 change greater than 2. So in this example, the differentially expressed genes, they have really big effects. So that's why I use log2 change greater or equal than 2. So people normally use greater or equal than 1, for example, and the p-adjustment is very smaller than 0.05. So you can change those cutoffs and kind of subjectively. And when you have too many, you want to filter down a little bit. So you use a little bit more stringent criteria. So if we do that, so, and this is all the differentially expressed genes that based on this cutoff. And we still get almost 450 genes here, okay? So once we do that, we can do pathway enrichment analysis, right? Once you get those differentially expressed genes. And there are 2 ways you can follow the cluster profiler tutorial, which is here. It is very comprehensive and one of the most highly packaged to do pathway enrichment analysis. So there are 2 different ways to do that. The first is the so-called over-representation test. So essentially you have the number of genes that you define that differentially expressed, and then you have multiple pathways that are human curated, and each gene set has a certain number of genes. then essentially just a hypergeometric test, okay, because you have this many genes that are differentially expressed, and then how many of those genes are in a certain gene sets, and then you can just calculate that in hypergeometric test. And let's load the library. And we need to first actually convert the gene symbols to untrans IDs. So we can use the cluster profile BITR function. This is the significant genes from symbol to untrans. And this is the database that we're going to use to map gene names to the untrans ID. So if we do that, take a look at, so we have the symbol mapped to the untrans ID. Also, we need to define the background gene sets here. So in this case, so all the genes that show up in our expression table, and I'm also filtered those genes with a 0 expression, and then we can take a look how many. So we have 14,000 genes as the back. So those are the genes that we use in the experiment. Similarly, we can convert that gene symbol to untrans ID. So if we take a look. Okay. So now we can do the enrichment. In this case, we'll first do a goal term enrichment, gene ontology enrichment analysis. So it defines the concepts and classes to distinguish between those concepts. And there are usually 1, 2, 3, 4 different categories. For example, MF stands for molecular function, CC stands for cellular component, BP stands for biological process. Okay, 1, 2, 3, actually 3 different categories. So in this case, we can use the enrich go function. And this is the genes that are differentially expressed, and we'll use the untrans ID and the universe, which is the background genes. We have that as well, and supply the data ontology will be biological process. Okay, use the BH methods for adjustment p-value, and those are the cutoff. Okay, so if we run that, it takes a couple seconds. Okay. Okay, it's done. So we can take a look at the results. So this is the enriched go terms. Okay. And we can load the library enrich plot, and then we can plot that. Show a bar plot here. Look at that. So those are the top enriched biological processes using the goal term enrichment analysis. So as you can see, the first one is angiogenesis and vascular development, and also response to oxygen levels. And you know, oxygen hypoxic condition can promote angiogenesis in cancer, and this enrichment analysis makes sense biologically to me. Okay, and then we can also try a dot plot. It's a different way to visualize it. Okay. Okay. So the p-value by color, this by the number of genes in that, the size of the dot means the number of genes in that. Okay, we can also use MSigDB, which is a very different gene sets that are curated from the group. And then we usually use the hallmark gene sets for cancer-related studies. And in this case, we can use this R package called MSigDB to download all the gene sets. So we can load the library. So you need to install it if you don't have it yet. And then we can just say, okay, species is human to download all the gene sets that is from human. Also takes a couple of seconds. Okay. Okay, it's done. If we look at it, so those are just a very tidy data frame with different columns here. You have gene symbol, ensemble genes, and then you have also the, so if you want to look at the full table here, you can do this. So you have the gene set name, gene set collections here, subcategory. Okay. So what we will do, we'll use collection of hallmark gene sets, and then select the gene set name and also the NCBI gene name, which is ensemble ID. So if we do that, you see there are two columns here. This is a gene set. We only subset, so that's why they're all prefixed with hallmark. And this is the on-chance ID name, okay, gene name. So with that, we can use the enricher function. Again, the significant genes, we supply them with on-chance ID. And this term to gene is this data frame that we will use. Okay, and then the universe is the background. Okay, let's take a look. And take a look at the result. And we see actually hypoxia is one of the top pathways that is enriched. So we can use the dot plot. Take a look. See, hypoxia is one of the most enriched. Of course, epithelium to mesenchymal transition, I think it's known that hypoxia can drive that process as well. Okay. So I showed you how to do overlap analysis, but there's another way to do pathway analysis. It's called gene set enrichment analysis. So in this case, you need to use all genes, and then you need to pre-rank those genes using certain metrics. So in the previous analysis, so we defined the cutoff and then only used the differentially expressed genes or significantly differentially expressed genes. So in this case, we'll use all the genes in the experiment and rank them, and then do gene sequence analysis. In the tutorial, the link that from the file, it talks about the differences between the overall representation analysis and the gene set enrichment analysis. And gene set enrichment analysis, that I think the paper was published in PNAS. I think that was enrichment analysis, PNAS, and it was cited by 50,000 times. So if you really want to understand, you can read this paper. Okay. Let's go back to here. Okay. Let's first rank those genes. But I first filtered those genes with 0 expressions here. And I'm going to use this sign, which is the sum of the log-to-fold change times minus a log10 p-value as the ranking matrix. Okay, so the most significantly upregulated genes will be in the front, and the most significantly downregulated genes will be in the end. Okay, so we just arrange that. Okay, so the gene list will be the rank stats, and the name of that gene list will be the on-chance ID. Okay, do that, and then take a look first. And something interesting here is that the first couple of them are infinity, because they have 0 actually p-value. So if you do that, it will give you error, okay? Okay, let me get it back. So that's why, if you look at this again, the data frame, the first couple of them are infinite because they have p-value 0. So let's just change them to arbitrary large numbers here. So if it's infinite, and then we just put them as 1,000. For example, it's just arbitrarily big number. So let's do that. And then recreate that gene list. And then if we look at the first couple of them, they become 1,000. It's just a way to avoid. Now we can run the GSEA function. So this is the gene list. Again, this is the matrix. And the name of that list, that value options ID, which is the gene ID. Again, we use that term to gene is that data frame for the whole. So we can do that. Okay. So we can take a look. Okay, we can visualize it using the GSEA plot and then specify the gene set. For example, you want to visualize. In this case, we're going to visualize the G2M, this one, the checkpoint, and also the hypoxia gene set. As you can see, this is called the normalized enrichment score here. So when it's minus, it means that it's downregulated, which is when it's positive, it means I can make the GSEA plot for both of them, and then we can plot them side by side, P1/P2. So as you can see here, okay, the G2M gene set is downregulated, and the hallmark hypoxia gene set is upregulated. So remember, so that how to read this figure. So the x-axis is here. Those are all the genes that are ranked, okay, from the most upregulated to the most downregulated. And those black bars are the genes in that gene set, okay? For example, for hypoxia, you have most of the genes in this pathway that are enriched in the front end of that gene pre-ranked gene list. And for the G2M checkpoint, you have most of the genes from this pathway, they actually show in the pre-ranked list, okay? But again, we rank the full gene list from the most upregulated to the most downregulated, okay? So this means hypoxia pathway is upregulated and G2M checked. Okay. So I have shown you end-to-end to how to do a single comparison, like hypoxia versus normoxia, and using the standard DC2 workflow. And this is nothing special, and everybody can follow the tutorial to do it. But I'm going to show you a really powerful skill to do multiple comparisons and using the map function from the purrr and use this powerful data structure tibble, which can contain a list column to avoid the repetition of your code. Okay, so in this case, we first make a data frame, and if you look at this, because we want to do, for example, 5 comparisons. Hypoxia versus normoxia under control, we have done that. But in this case, we want to do more comparisons. For example, knock out this versus control under normoxia condition, and knock out the other gene versus control under normoxia condition. But we can also do that under hypoxia condition, right? So in this case, 5 different comparisons. So we first actually create this. using the samples that we're going to use, okay, for those 5 comparisons. And change this as a data frame and then add the row names using the sample ID. Okay, so this is what we get eventually. Okay, so to do that, we first need to write a function to do a one comparison. Okay, so we write a function, and this the counts, and this the design table, and the contrast name, for example, rna-seq2, this function, and then we will filter this design table to the contrast, the comparison that we're going to use, and subset the count matrix using only the samples that are related to the comparison. Do the filtering of the small counts of the genes, and then then return the differential expressed gene table. Okay, so we can run that. Okay, so we can test a single comparison. So in this case, rna-seq2. This is the raw counts, and this is the design table we just created. But the contrast name here, we're going to just compare the hypoxia versus normoxia for the control guide RNA. So if we do that, and then take a look at the differential expressed gene table, this is the same as what we did manually for one comparison, the results. So those two are the same. Okay, 1477 3G. So now that we verified that a single function works for one comparison, now we can apply the same function to other 4 different comparisons. Okay, we should actually keep everything in a single table or tibble. So let's create a new data frame called results.df. And then in this data frame, we are defining the contrast nominator versus denominator. So in this case, this versus this, and this versus this. Okay, so what we can do, so this the original data frame like this. I'm going to deploy a mutate function and add another column called results, and I'm going to map this map2 function, so this x and this y parameter options here, and then we will say rna-seq2, this is the function we define here, and then we have these. Let's see here. Design table is design table, but the condition here will be .x will be filled in by this column, and .y will be filled in by this column. Okay, so if we do that, it's going to do differential expression analysis for 5 different comparisons. Okay, so let's take a look at this new data frame. So you remember this data frame we started kind of constructed manually, but we added a new results column, and it contains D-seq results table objects, right? And we can actually access individual D-seq2 results by using the $, access this column, and because it's a list column, it's a list of D-seq results, and we can use the double bracket number to access each of them. Okay, and you can do that to look at the second. Okay, this is very cool, right? This is very cool because we can actually convert this as a data frame. So this is a D-seq2 result. So we can mutate again, and then add another column, and then map the results column, this column, and change them as data frame. Okay, and then let's remove this results column first. And if you look at it again, it will show you now we have the results 2 column, but this column contains a list of data frame instead of a list of RNAseq object. So we can actually unnest it into a full data frame. For example, you use the tidyr unnest. Column will be this column. If we do that, it actually unnest that into a full data frame. So now we have this results from the RStudio table showing here. The nice thing about this is this is a tidy data frame, right? And it is easy to make any figures when you have a data frame using ggplot2. Okay, so now we're just adding a new, for example, you do this step by step, build the pipes, and mutate and add another column, and then just paste the contrast denominator versus the contrast denominator. And now you use ggplot2 function, and then x equals log to fold change, and the y-axis will be the minus log 10 p-value. So this is a commonly used volcano plot. But in this case, we can use the facet wrap and then split that plot by comparisons, which we defined just by concatenating those 2 columns together. So let's take a look. So you'll see instantly 5 different volcano plot by a single function, right? Because you did 5 comparisons, and you can make 5 volcano plot in one call. This is very powerful, actually. Okay. And this is the column that the comparison, and the new column that we constructed. Okay, so you know the comparison. Okay, let's actually find differentially expressed genes for each comparison by using some cutoff, for example. So in this case, we're defining a new function called filter significant genes, and this differential gene expression expressible change to data frame and filter by some arbitrary colorful here. Okay, and then extract the gene names and return the gene names. If we do that, and we can test for one anorexia, and then use that gene function, and it will tell you the first couple of different genes, significant genes. So in the same way, we can now remember this is the data frame we have, and then we can mutate and add another column here called sig genes, and map this function which we just defined to this column. Okay, so every element of this list will be filtered. Okay, so if we do that, take a look. Now you have another column called significant genes, and it's a list of characters. Okay, so you have 502 significant genes past that filter, this cutoff. Then you have 192 for this comparison. Okay. So let's keep going and to do a gene set over representation analysis for each of the comparisons here. So again, we have that gene set, so 2 columns, gene set name, and then the on-chance ID. So if we do a single comparison here, you can do enricher. This is how you do for a single comparison. But what you can do now, you can apply this enricher function to every element of that colon. Okay, so remember, this is the data frame we have, and we're going to mutate to add another column here called hallmark result, then map that function, enricher function, to this column. Okay, so this will be the x, so dot x will be the significantly expressed genes, and this is the term to gene data frame, and the universe will be just the background gene for all the gene symbols. Okay, so let's do that. And if you take a look, and then you have, okay, let's see, it says now, so something is wrong here. Let's see. Okay, let's redo it. No genes mapped. Oh, so we need to actually define a new M2 ID, and in this case we're using the symbols. So in this case I'm actually redo this, but now I'm using the gene symbol. Okay, let's redo that and take a look again. Now we have two columns, gene set name and gene symbol, but previously I used gene set name and also the NCBI name, which is ontrans ID. So as long as you have consistent IDs, for example, you use ontrans ID here, and you also use ontrans ID here, and then in this data frame you also use ontrans ID. But in this case, I'm using the gene symbol, and then here, those are also gene symbols. So I will swap them to be gene symbols here. Okay, if you do that, now take a look. Now we have the enrichment results here. So now you see the pattern. You can keep adding the results into a new column, because table can contain a list column, and the list can contain any objects, even the ggplot2 figures. Okay, let me show you. Now you can actually mutate and another column called dot plots and a map. Okay, essential map in this column. So every element of this list, and apply the dot plot function. Look at that. Take a look at this. Now you have this dot. So you can take a look of them by use $sign to access this column, and then use double bracket to access the element of a list. And see, you have the dot plot here. Okay. And then you can look at the second one is there's no term enriched, so this is not. And then third comparison here, there's only single one at the end of a single mode transition, and and then the fourth one also a single pathway that is enriched. And you can see how powerful is that. Now you are keeping everything into a single data frame or table. You can have all the enrichment results, all the dot plots, which is ggplot2. You can also modify the ggplot2 using the mutate function again. And for example, you want to add a title of for each of the enrichment plot. You can do that using the mutate function. And then you need to define a function to, because this is ggplot2, you can add plus gg title, for example, add a title. Okay. And lastly, you can actually save those plots into file by this pwalk function. Okay, and then you can define and see. So pwalk make multiple if you want parameters here, and then C is the dot plots. Then you can safely use those A versus B. Essentially, is A versus B, and then save that plots into file. Okay, and that's it for today, and hopefully you see how powerful the map function is, and also how useful the list column in table is. And once you are getting used to the map function, you can write fewer and fewer loop code in your R code. And I hope you find it helpful, and happy learning. I will see you next time. And make sure you subscribe to the Chatnomics channel and forward it to your friends if you find it helpful for you. Sharing is caring. I'll see you next time.